Hello everyone, today we will read Chapter 2 Pharmacokinetics, Membrane Transport, Absorption and Distribution of Drug. Competency covered is describe absorption and distribution of drugs. Pharmacokinetics is the quantitative study of drug movement in, through and out of the body. The intensity of response is related to concentration of drug at the site of action, which in turn is dependent upon the pharmacokinetic properties. Pharmacokinetic considerations therefore determine the routes of administration, dose, latency of onset, time of peak effect, duration of action, and frequency of administration of the drug. The overall scheme of pharmacokinetic processes is depicted in Figure 2.1: absorption, distribution, and storage and excretion. Drug in dosage form, released drug in the solution, which goes in the blood where the free drug binds to protein. It gets metabolized. There occurs biotransformation. In the storage tissue, it is converted into free and bound form, and at the site of action again there is equilibrium between free and bound form. All pharmacokinetic processes involve transport of a drug across the biological membrane. Abbreviations: ABC ATP binding cassette, AVD apparent volume distribution, BBB blood-brain barrier, BCRP breast cancer resistance protein, CSF cerebrospinal fluid, CYP cytochrome P450, DAT dopamine transporter, GLUT glucose transporter, GNT glyceryl nitrate, 5-HT 5-hydroxytryptamine, MAO monoamine oxidase, MDR multi-drug resistance, MIC minimal inhibitory concentration, MRP multi-drug resistance associated protein, NET norepinephrine transporter, OATP organic anion transporting polypeptide, OCT organic cation transporter, PGP P-glycoprotein, SERT serotonin transporter, SGLT sodium glucose transporter, SLC solute carrier, SSRI selective serotonin reuptake inhibitor, VMT vascular monoamine transporter 2. Biological membrane is a bilayer about 100 angstrom thick. It is a phospholipid and cholesterol molecules. The polar groups glyceryl phosphate attached to ethanolamine or choline or hydroxyl group of cholesterol are oriented at the two surface, and the non-polar hydrocarbon chains are embedded in the matrix to form the continuous sheath. This imparts high electrical resistance and relative impermeability to the membrane. Extrinsic and intrinsic protein molecules are absorbed on the lipid bilayer. The intrinsic protein extends through the full thickness of the membrane, surround fine aqueous pore. Paracellular space or channel also exists between certain epithelium or endothelium cells. Other absorbed proteins have enzymatic carrier, receptor, or signal transduction properties. Drugs are transported across the membrane by passive diffusion and filtration. Specialized transport. So, here is the illustration of organization of biological membrane. This is intrinsic protein, which has the pore, hydrophilic and hydrophobic part of the lipid, which is phospholipid, then extrinsic protein and carbohydrate attached to the protein. This is the CSF side, this is cytosol side. Now let us see passive diffusion. So the drug diffuses across the membrane in the direction of its concentration gradient, high to low, the membrane playing no active role in the process. This is the most important mechanism for majority of drugs. Drugs are foreign substances known as xenobiotics, and a specialized mechanism are developed by body primarily for natural metabolites. Lipid-soluble drug diffuse by dissolving in the lipodial matrix of the membrane. Here is the illustration of passive diffusion and filtration across lipodial biological membrane with aqueous pore. Here there is high concentration, here there is low concentration. So in the passive diffusion, there occurs the movement from higher to lower concentration, which is these red particles are lipid soluble drugs, and these blue ones are non-lipophilic, that is non-lipid soluble drugs. So through filtration, aqueous pore or paracellular spaces. This is one cell, this is the gap, then here there is another cell. So through the gap between two cells, which is referred as paracellular space, there occurs the transport of non-lipophilic substances, as they are not permeable through the membrane. Now the rate of transport being directly proportional to the lipid water partition coefficient of the drug and concentration gradient of the drug across the membrane. Influence of pH. Most drugs are weak electrolyte and their ionization is pH dependent. In contrast to the strong electrolytes that are nearly completely ionized at acidic as well as alkaline pH. The ionization of weak acid is given by the equation pH equals to pKa plus log of A negative by HA, where pKa is negative logarithm of acidic dissociation constant of weak electrolyte. If the concentration of ionized drug is given equal to the concentration of unionized drug, then their value will be 1. Since log 1 is 0, then under such conditions, pH will be equal to pKa. Thus, pKa is numerically equal to pH at which the drug is 50% ionized. If pH is increased by 1 scale, then log of A anion divided by HA is 1, or A negative by HA is 10, because log 1 is 10. Similarly, if pH is reduced by 1 scale, then it will become 1 by 10. Thus, weakly acidic drugs which form salts with cation, for example, sodium phenobarbitone, sodium sulfadiazine, potassium penicillin 5, etc. ionize more at alkaline pH. And 1 scale change in pH causes 10-fold change in the ionization. Weakly basic drugs which form salts with anion, for example atropine sulfate, ephedrine HCl, chloroquine phosphate, etc., conversely ionize more at acidic pH. Ions, being lipid insoluble, do not diffuse, and the pH difference across a membrane can cause differential distribution of weakly acidic and weakly basic drugs on both sides. Here the pH is 5 and here it is 7. At pH 5 the drug is unionized, and at 7 as well it is unionized, which is separated by a membrane. Ionized at 1, unionized at 10, then again ionized at 100. So total here the pH is 10 plus 1 11 and here 100 plus 10 110. So in figure 2.4 the influence of pH difference on both the side of the biological membrane on a steady state distribution of weakly acidic drug with pKa 6. As the difference between the ionization is the same, so both are becoming ionized. Clinical importance of the consideration are acidic drugs, for example aspirin, which has pKa of 3.5, are largely unionized at acid gastric pH and are absorbed from the stomach, while basic example atropine, which has pKa of 10, are largely ionized and are absorbed only when they reach the intestines, which has basic pH. The unionized form of acidic drugs, which crosses the surface membrane of gastric mucosal cell, revert to the ionized form within the cell, which has the pH 7, and then only slowly pass to the extracellular fluids. This is called ion trapping, that is, a weak electrolyte crossing a membrane to encounter a pH from which it is not able to escape easily. This may contribute to gastric mucosal cell damage caused by aspirin. C. Basic drugs attain higher concentration intracellularly, pH 7 versus pH 7.4 of plasma. D. Acidic drugs are ionized more in alkaline urine, do not back diffuse in the kidney tubules, and are excreted faster. Similarly, basic drugs are excreted faster in urine is acidic. Lipid-soluble non-electrolytes, for example ethanol, diethyl ether, radially cross biological membrane and their transport is pH independent. Filtration. Filtration is the passage of drugs through the aqueous pores of the membrane or through the paracellular spaces. This is faster if hydrodynamic flow of solvent is occurring under hydrostatic or osmotic pressure gradient, for example across the most capillaries including glomeruli. Lipid-insoluble drug crosses the biological membrane by filtration if their molecular size is smaller than the diameter of the pores. Majority of the cells, intestinal mucosa, RBCs, etc. have very small spores of 4 angstrom, and the drugs with molecular weight less than 100 or 200 are not able to, more than 100 or 200 are not able to penetrate. However, capillaries, except those in the brain, have large paracellular spaces of 40 angstrom, and most drugs, even albumin, can filter through these. As such, diffusion of drugs across the capillary is dependent on the rate of blood flow through them rather than the lipid solubility of the drug or pH of the medium. Now we will see specialized transport. This can be carrier mediated or by vesicular transport, which is endocytosis, exocytosis. First is carrier transport. All cell membrane express a host transmembrane proteins which serve as a carrier or transporter for physiologically important ion, nutrient, metabolite, transmitters, etc. across the membrane. At some sites, certain transporters also translocate xenobiotics, including drugs and their metabolites. Transporters combine transiently with their substrate, ion or organic compound under a conformational change, carrying the substrate to other side of the membrane where the substrate dissociate and transporter return back to their original state. Carrier transport is specific for a substrate or the type of substrate. Example, organic anion substrate saturable, competitively inhibited by analogues which utilize the same transporter and is much slower than the flux through channels. Depending on a requirement of energy, carrier transport are of two types. Facilitated diffusion. The transporter belonging to the superfamily of solute carrier SLC transporter operate passively without needing energy and translocate the substrate in the direction of its electrochemical gradient, that is from higher to lower concentration. See figure 2.5a. It merely facilitates permeation of poorly diffusible substrate, example the entry of glucose into muscles and fat cells by the glucose transporter GLUT4. Second is active transport. It requires energy, is inhibited by metabolic poisons and transports the solute against its electrochemical gradient, which is low to high, resulting in selective accumulation of substances on one side of the membrane. Drugs related to normal metabolites can utilize the transport processes meant for these. Example, levodopa and methyl-dopa are actively absorbed from the gut by aromatic amino acid transporter. In addition, the body has developed some relatively non-selective transporters like P-glycoprotein (PGP) to deal with xenobiotics. Active transporter can be primary or secondary depending upon the source of driving force. A primary active transporter, energy is obtained directly by hydrolysis of ATP. See figure 2.5b. The transporter belonging to the superfamily of ATP binding cassette ABC transporters whose intracellular loops have ATPase activity. They mediate only efflux of the solute from cytoplasm either to extracellular fluid or into the intracellular organelles, endoplasmic reticulum, mitochondria, etc. Encoded by multidrug resistance 1 MDR1 gene, PGP is the most well-known primary active transporter expressed in the intestinal mucosa, renal tubules, bile canaliculi, choroidal epithelium, astrocyte, foot processes around the brain capillaries, the blood-brain barrier, testicular and placental microvessels, which pump out many drugs, metabolites, and thus limit their intestinal absorption, penetration into brain, testes, and fetal tissues, as well as promotes biliary and renal elimination. Many xenobiotics which induce or inhibit PGP also have a similar effect on the drug metabolizing isoenzyme CYP3A4, indicating their synergistic role in detoxification of xenobiotics. Other primary active transport of pharmacological significance are multidrug resistance and associated protein 2 MRP2 and breast cancer resistance protein BCRP. Low concentration, high concentration, poorly diffusible substrate, SLC transporter. So, illustration of different types of carrier mediated transport across the biological membrane: ABC ATP binding cassette transporter, solute carrier transporter, and M is membrane. Figure A, which we have seen, is facilitated diffusion. The carrier SLC solute carrier transporter binds and moves poorly diffusible substrate along its concentration gradient high to low, does not require energy. Then figure B, primary active transport. The carrier ABC drives energy directly by hydrolyzing ATP and moves the substrate against its concentration gradient from low to high. Then C, is the symport. The carrier moves the substrate A against its concentration gradient by utilizing energy from downhill movement of another substrate B in the same direction. Then D. Antipode. The carrier moves the substrate A against its concentration gradient and is energized by downhill movement of another substrate B in opposite direction. PGP inducer are rifampicin, phenytoin, clotrimazole. PGP inhibitors are verapamil, diltiazem, cyclosporin, hydrocortisone, propranolol, quinine. MRP2 inhibitors are probenecide, ritonavir, furosemide, lamivudine, nevirapine, cidofovir. Now, secondary active transport. In this type of active transport effected by another set of SLC transporter, the energy to pump one solute is derived from the downhill movement of another solute, mostly sodium. When high concentration gradients are such that both the solute move in the same direction, which is shown in figure 2.5c, it is called symport or co-transport, but when they move in opposite direction, which is shown in figure 2.5d, it is termed as antipode or exchange transport. Metabolic energy from hydrolysis of ATP is spent in maintaining high transmembrane electrochemical gradient of the second solute, generally sodium. The SLC transporters mediate both uptake and efflux of drugs and metabolites. The organic anion transporting polypeptide OATP and organic cation transporter OCT are highly expressed in liver canaliculi and renal tubules. They are secondary active transport important in the metabolism and excretion of drugs and metabolites, especially glucuronides. The sodium chloride dependent neurotransmitter transports for norepinephrine, serotonin, dopamine, NET, SERT, and DAT are active SLC transporters that are target for action of drugs like tricyclic antidepressants, selective serotonin reuptake inhibitors, SSRIs, cocaine, etc. Similarly, vesicular monoamine transporter VMAT2 for adrenergic and serotonergic storage vesicle transport catecholamines and 5-HT into the vesicles by exchanging their H plus ions and is inhibited by reserpine. The absorption of glucose in intestine and renal tubule is through secondary active transport by sodium glucose transporter SGLT1 and SGLT2. Carrier transport both facilitate diffusion and active transport is saturable and follows Michel-Menton kinetics. The maximal rate of transport is dependent on the density of transporter in particular membrane and its rate constant, that is, the substrate concentration in which the rate is half maximal is governed by its affinity for substrate. The genetic polymorphism can alter both the density and affinity of the transporter protein for different substrates and thus affect the pharmacokinetics of the drug. Second is vesicular transport, endocytosis, exocytosis. Certain substrates with very large or impermeable molecules are transported inside the cell, endocytosis, or extruded from it, exocytosis, by enclosing their particles in tiny vesicles. A binding protein located on the membrane complexes with substances and initiates vesicle formation. The vesicle then detach from the membrane and may remain stored within the cell, or it may release the substance in the cytoplasm, or it may move to opposite membrane, fuse with it to release the substance across the cell. Exocytosis. Vesicular transport is applicable to proteins and other big molecules and contribute little to the transport of most drugs bearing few, like vitamin B12, which is absorbed from the gut after binding to intrinsic factor, a protein. Most hormones, insulin, etc., neurotransmitters like noradrenaline are secreted and released from the cell nerve ending by exocytosis. Activation of the secretory cell nerve ending prompts fusion of the storage vesicles to the surface membrane, followed by extrusion of its content into the extracellular spaces. Illustration of vesicular transport, endocytosis and exocytosis. In endocytosis, the large molecular particle P binds to binding protein B on the surface of the cell. The membrane invaginates to form a vesicle which nicks off, and the vesicle may remain stored within the cell, or it may disintegrate to release substance in the cytoplasm, or be extruded across the cell by exocytosis. Exocytosis, the particle or the transmitter hormone stored within the intracellular vesicle, generally as a complex with a storage protein, is secreted by exocytosis. On activation, vesicle translocate and fuse with the membrane. All the content of the vesicle are then poured out of the extracellular space. This is exocytosis. This is exocytosis. This is endocytosis.